Hello everyone and welcome to this audio podcast today. This audio podcast as part of a Serology and Squil. This audio podcast as part of a Hustle and Serotype. This audio podcast as part of a Defcon premiere. This audio podcast as part of a Storm Robotics production. Continuing on looking at immunobiology and stratifying receptors. Now going into some further details regarding or relating to laboratory investigations. So additionally, early research had informed that activated CD3 positive lymphocytes which express chemokine receptors CCR5, CXCR3, and CXC3CR1 are highly responsive to chemotactic factors produced by proximal tubular epithelial cells, or PTEC, which in turn have been stimulated by pro-inflammatory cytokines. Previous research had also informed of how a recruitment of both T cell receptor interplay, and then the ligation of high affinity interleukin 2 receptor for full T cell activation, for full T cell activation. Early laboratory research had also demonstrated through blockade studies that chemotaxis for PTEC is mainly attributable to ligation of CCR5 and CXCR3. Ligation of CX3CR1 has an additional effect. Investigations have also demonstrated using a T cell culture system that all activated T cells that express CCR5 and also CXR3 positive, indicating that these cells may be attracted through either receptor. Investigators had demonstrated that virtually all T cells at sites of non-renal or non-kidney human inflammation have prominent mononuclear cell infiltrates that were CXCR3 positive. One team also informed that up to around 40% of freshly isolated T cells express CXCR3, but cells also migrate towards IP10, CXCL10 after activation. However, it was earlier research that also identified just over 20% of freshly isolated T cells also express CXCR3, and 15% had expressed CCR5, but no activation by anti-CD3 antibody, and approximately 90% of cells express both receptors accordingly. That draws this audio podcast to a close. This audio podcast is part of the Serology and Squil. This audio podcast is part of a Hustle and Serotype. This audio podcast is part of a Defcon premiere, and this audio podcast is part of a Storm Robotics production.